Hello everyone, my name is David Harmon. I'm a business development manager here at Synopsys Simpleware, and today I'm extremely proud to introduce Professor Amit Geffen of the University of Tel Aviv. Amit is a very renowned researcher at the University of Tel Aviv and professor, and he's joining us today to provide a webinar based on his use of computational models within his research. And his webinar today will focus on computational models in wound care research, and his title is The Key to Innovation. And today Professor Amit Geffen will really focus on how these technologies have been used within the COVID-19 pandemic to help alleviate problems that have arisen. We're extremely proud to have Amit with us today. Before I pass over to Professor Geffen, I will introduce the Simpleware software and provide some introductory slides for Amit too. But welcome everyone, and thank you very much for joining us today. So as I mentioned, a short introduction about the Simpleware product group at Synopsys. Really, our focus is the development of high-end 3D image processing software, and wonderfully, Amit today will show you how he and his group, his research group at Tel Aviv University, have really used the software in its best way to help biomechanical research. So as a software provider, obviously we have dedicated sales, support and service teams around the world. And we work with customers in life sciences, as you may have gathered from this webinar today, but also in broader industries too, for instance, materials and more heavy industries and high value part generation and development. So today you'll understand that our applications are really focused heavily towards biomechanics, and if we split those applications up into two different areas, we have more clinical sides and more applications towards product interaction with the human body. So you can see from the examples on screen the sort of areas we work in, modeling the human body and protective gear together to understand the effectiveness of that equipment. On the top left hand side there, and you can see that we also work in the area of wearable electronics, looking at the interaction between the device and the human body, and the same with other clothing, as you can see on the bottom there. On the clinical side, we have applications in the area of surgical planning, for instance, and implant development, but also on physiological simulation, so more biomechanical simulation, understanding the physiology of human beings and understanding how changes in environment can affect the human body, and as Professor Geffen will show you later on, how the software can be used to analyze how stresses are generated and what that means for people in care. And we can also use our software for developing and creating different designs for 3D printing, or indeed 3D printing human anatomy itself, for aiding the diagnostic process or education process of a variety of groups of people. So looking on the technical side of our software, what does it do? Really, our focus can be seen in these hexagons here. So we have capabilities for importing and visualizing data, image data, 3D and 4D, we should say. We could have a large number of different tools for segmenting that data, ranging from manual all the way to extremely automatic with our machine learning based tools we've implemented. Obviously, once we have that segmentation, we can go through into measurements and analytics, understanding that data quantitatively. We can also start to integrate CAD with the image to understand how implants are interacting with the human body. We can then generate models for a variety of different applications, including simulation and design. And then tying all of this together, we have a very open scripting API and interface that allows you to link all of the different aspects of this workflow together using very well-known scripting languages such as Python. Okay, so that's the short introduction of the Simpleware software. Now, for the really exciting part, I'm going to hand over to Professor Amit Geffen, and he, as I mentioned earlier, he's going to show you all of the different applications of his group in the area of computational modeling and for biomechanics and its applications to wound care research, with a focus on some of the things the group has been doing to have a positive impact during the COVID-19 pandemic. So an introduction to Professor Amit Geffen. So Professor Geffen is a full professor within the Department of Biomechanical Engineering at the University of Tel Aviv, and he's also the Berman Chair in Vascular Bioengineering. His research topics are really focused on normal and pathological effects of biomechanical factors on the structure and function of cells, tissues, and organs, and his research has emphasized on how these findings and applications can help in wound care research. So with all of this work, he's published more than 270 articles in peer-reviewed journals and edited multiple books on mechanobiology, cell and tissue biomechanics, and applications mostly around wound prevention and treatment. So we're really excited to welcome you here today, Amit, and really looking forward to your presentation. We're really passing this on to you and really looking forward to seeing your work. Thank you very much, Amit.
Hello, my name is Professor Amit Geffen. I'm a professor of biomedical engineering at the Faculty of Engineering of Tel Aviv University. And I'd like to thank the Synopsys team for the introduction and for the opportunity to give this webinar today. The topic of my webinar is how to use computer models in wound care research, which is my research field. Biomedical engineering, by definition, is an integrated field. It integrates knowledge from different disciplines, the traditional sciences, physics, biology, mathematics, or chemistry, with the more advanced or modern sciences such as computer science or materials engineering. And you are well aware of the fact that biomedical engineers have changed our world. We are now implanting hip or knee joints, artificial ones, routinely all over the world. Same goes with cardiovascular implants, where we routinely replace heart valves or implant stents, and dental implants as well. All of that has been made possible by this integration of disciplines, but more than that, the ability to use basic science, fundamental science, to understand the etiology of these conditions, of these diseases, and then to mitigate the specific factors in the etiology and the pathophysiology of the disease that cause the deterioration in the quality of life or endanger life itself, is what made these interventions successful, these innovations successful. And I think that the same can be done in wound care, and bioengineering has a lot to contribute to wound care, using similar methodologies, approaches to the ones that were used to develop the breakthroughs that we see in the slide here. And this is my research agenda. My career is dedicated, devoted to understanding the etiology of chronic wounds, with emphasis on pressure ulcers or diabetic foot ulcers, but also other types of injuries, and then developing the most effective means to either prevent or preferably prevent, and if we can't prevent, then treat those wounds in the most effective way that addresses the fundamental factors in the etiology. This is what I do in my research. For example, in the context of pressure ulcers, pressure injuries caused by medical devices, investigating the etiology and then changing the design of devices so that they better fit the skin and the underlying tissues, and by matching the device properties to the tissue properties so that they don't inflict these devastating injuries that we see here caused by devices of all types, we engineers can do a lot to change the world. So stopping pressure injuries or pressure ulcers is the ultimate goal, but in order to reach that goal, as I've said, we need to understand the etiology. And the etiology of pressure ulcers or injuries is fundamentally a biomechanical one. They're caused by either body weight forces or external forces. It can be static body weight forces that deform and distort tissues, but not only tissues, but also everything that is contained within tissues, including the basic living unit in the human body, which is the cell. So cells are distorted, cells are deformed, and everything within the cells is also deformed by body weight forces. The cell organelles are distorted, and that distortion eventually, after a certain period of time, starts to affect their biological function. And that not only relates to static body weight forces, such as during sitting or lying, it also connects to the etiology of, for example, diabetic foot ulcers, which are caused often by dynamic body weight forces. But again, these dynamic body weight forces distort cells and tissues and thereby eventually cause the loss of cell and tissue viability. Another example of forces that are or can be sustained and that lead to tissue injuries are those applied by medical devices. These are external forces as opposed to body weight forces, either static or dynamic, but external forces are also harmful or can be harmful if being too prolonged or too intense. And we can all think of the classic example of a CPAP mask, which is now being used often in intensive care or other care facilities to treat patients with COVID who develop respiratory distress. And these masks apply continuous loading on facial tissues, on skin, and eventually cause tissue damage. In the pandemic, we also saw a new type of device-related pressure ulcers, which are pressure ulcers caused to healthy subjects, healthy individuals, medical workers. And these healthcare professionals who use their face mask or respirators for long times develop facial injuries, which I have reported and investigated substantially during the pandemic. And my conclusion was that they share the etiology of the pressure ulcers seen in patients, particularly the device-related pressure ulcers seen in patients, in the sense that they compromise cell and tissue function through sustained deformations. And let me take you into a little bit more of detail in how that happens. When cells are deformed, the entire cell structure is deformed. That includes the cytoskeleton, the skeleton of the cell. That includes the envelope of the cell, the walls of the cells, which are the plasma membrane. And you can think about it as in kind of an analogy. Think of this poor, tortured person on the torture rack. Now, this person is subjected to sustained forces applied by the torture rack. And what these sustained forces do is that they distort the skeleton of that person. And the skeleton is the protective element, protective structure in the body that is protecting all the internal vital organs. And once the skeleton is damaged, the vital organs would be damaged as well, and the person dies in pain. And basically, the same happens in cells that are subjected to sustained deformations. The cytoskeleton is gradually being damaged. That damage to the cytoskeleton causes it to be less able to support the other cell structures, particularly the plasma membrane of the cell, that then develops sporation sites, small holes, gaps in the continuum of the plasma membrane that now allows transport, which is uncontrolled. So biomolecules that previously had to go through all these well-controlled gates along the perimeter of the cell, along the wall of the cell, such as ion channels or endocytosis mechanisms, or all these sophisticated mechanisms that the cell acts to maintain its homeostasis and control precisely what goes in and what goes out. These are not that they're not functional, but they're ineffective because there are these pores through which everything can now flow, and so the cell loses control over the influx and the outflux, or essentially the homeostasis of the cell body, which is essential for life, and the cell very quickly dies within minutes. And this is really the beginning of how a pressure ulcer or pressure injury forms. But as I said, this is just the beginning, because once these first cells have died because of the exposure to deformation, the body activates the immune system as the first response, and tries to recruit white blood cells to target that cell damage site and migrate into it in order to prepare the grounds for tissue repair. And that requires that the white blood cells leave the vasculature and cross over to the extracellular space, and so that they can migrate into the damaged site. While that happens, the blood vessels become leaky, permeable, and so fluid, plasma basically, escapes from the vessels and accumulates at the interstitial space. And we all know that as edema, which is what happens whenever you get a tissue injury of any source. Can be a thermal insult, a mechanical insult, a chemical, pathogen, biological insult. The immune system is recruited and edema is formed. That causes, in turn, a rise in the interstitial pressures. Now, think about this person with the CPAP mask that we have mentioned before. The facial tissues are in a sandwich, basically, between that CPAP mask and the rigid skull, and they cannot swell as in the swelling process that we are all familiar with when there is edema. So basic physics tells you that in that condition, interstitial pressures would rise very sharply, because if you cannot increase in volume, the pressure increases. And that obviously causes further cell distortion, further deformation, further poration of the plasma membrane, and further cell death, now fueling the inflammation again. And this becomes a vicious cycle for cells and then for tissues, and ends up as massive tissue damage or death, as you see on the right-hand side of the slide, for this loss of facial tissue at the nasal bridge, which is classic in CPAP masks or CPAP mask users, and it's very common as well. These types of damages are seen after as few as 2 hours of use. It all starts with the deformation. It's really the deformation which is the primary cell killer, which is why I'm always using this logo or motto, if you wish, that deformation is a cell killer. This is a computer simulation that demonstrates the etiology of what happens when a cell dies, and then basically the other cells are left to deal with the mechanical loads that are still there, are still sustained, but with fewer cells to support them. And so each of the remaining cells now needs to support more or a greater share of the mechanical loads, which of course implies that the likelihood that it will die next because of the exposure to mechanical loads increases. And to me, that always reminds me of my basic training in the Israeli army, where we used to do these stretcher marches with 4 soldiers carrying a simulated wounded soldier. And you cannot, in this type of training, you cannot leave the stretcher, because obviously the wounded soldier will fall. And so if something happens that one of the soldiers becomes fatigued and drops, the other 3 soldiers, the remaining soldiers, must carry the stretcher. And that's basically what happens here. The other cells must carry the load, and if other cells die, then the remaining cells still need to carry the loads. And so this is an escalating process where more and more cells are dying, and the others are at a greater risk. And from an engineering perspective, this etiology can be described using these 3 components that dictate together or form the damage etiology: the direct deformation, causing the loss of homeostasis to the cells; the inflammatory response, now increasing the pressure and deforming cells more; and finally, when pressures, interstitial pressures are high enough, you can also expect ischemia, ischemic damage, due to the obstruction or even occlusion of some of the blood vessels that now starve tissues from a biochemical perspective as well. So not only that their mechanical stress is there, now the chemical stress, the biochemical stress, is also building up. And if you add a disease onto this, such as diabetes, for example, and I've chosen diabetes to start with to show you that it's not only COVID, where you have an impairment or a disorder that affects any of these components, etiological components. For example, if you have a specific anatomy that causes increased tissue deformations, or if you have an impaired inflammatory response, such as in diabetes, which causes higher vascular permeability, leading to a greater extent or spread of the edema, then obviously that component in the damage cascade would be escalated, and the overall result would be more intense damage. In diabetes particularly, there is also this lower tissue repair capacity because there are fewer white blood cells and other tissue repairing cells due to the compromised perfusion in these patients, which is why pressure ulcers are so difficult to heal in diabetes. But the most important point is that this is very individual. And so what you see here is just the trends of the effects, but in any person, this would look differently depending on their specific age and health condition, or any medications that they may take at the time, and so on and so forth. So this is a much more complex description of the damage cascade with respect to what we used to know in the past as just ischemia, as just blood flow. And it all starts with the exposure to the deformation. So we as biomedical engineers have now a more targeted, specific task of looking at these tissue deformations, and both superficially at the skin and in deeper tissues, and finding ways to lower them or spread them better, make them redistributed in a sense that would not load cells and tissues to a critical level. And now comes COVID as a good example for explaining this concept, because in COVID, if you think of an ICU where a person is subjected to many different types of medical devices which contact their skin, and is also lying in bed for a long time, the issue of deformation becomes critical, because a lot of the body's surface is now subjected to sustained deformations, either due to body weight or due to the forces applied by medical devices. And so here is an excellent opportunity for using computer modeling to study these aspects. For example, a paper that we've published on prone positioning, which is one of the fundamentals of care with respiratory distress syndrome. But prone positioning causes very high tissue loads, particularly at the chin and at the forehead, which may compromise tissue viability there through all these mechanisms that I've just described. Now, with this computational description, you can now look at different solutions such as materials, positioners, prophylactic dressings, and so on, and determine their effectiveness quantitatively in a standardized manner so that different approaches for prevention of the injury can be evaluated. This is critically important at times of pandemic where there's no time or no capacity to do randomized clinical trials or basically any clinical trials. Another nice example of using computer simulations is to look at endotracheal tubes, a device that has not been changed for many, many decades, is known to cause device-related pressure ulcers on the lips, as you can see, or facial tissue in general, as you can see at the right-hand upper part of the slide. And we have studied the function of these devices in terms of the tissue loads that they induce, again, coming with some practical solutions with regards to the repositioning of the device or potential other means to alleviate the loads. Coming back to the CPAP mask. Now, here is application of computational modeling to determine these tissue loads quantitatively, again, so that materials, prophylactic dressings can be evaluated with respect to their direct effect on tissue integrity, or at least the loading state that can then be correlated with the likelihood of tissue damage. And another example on evaluating personal protective equipment, this time from a medical workers aspect. Now here is a specific example of looking at tissue loads, facial tissue loads, skin tissue compression when the mask is used with a lubricant versus without a lubricant, again, allowing to determine the specific reduction in the intensity of tissue load. This kind of describes the not only the vicious cycle of device-related pressure ulcers or pressure ulcers in general, but also the role of computer modeling in looking at the triggering event, which is the exposure to tissue deformation, not to mention, again, the circumstances that COVID has brought of intensifying that exposure to tissue deformations by use of medical devices, the prone positioning, and the effects on the other components of the vicious cycle, such as the cytokine storm that affects the inflammation, and the vascular effects on coagulation and obviously oxygen saturation. All of that not only connects to the etiology, but also to the prospects of how we can use computer modeling in order to study tissue health. So, to summarize, computer modeling-based research, particularly in pandemic times, is very effective because it allows identification of materials and methods to protect tissues where the body is exposed to sustained deformations, either due to body weight forces or due to medical devices. And since the regular route of clinical trials was disrupted during the pandemic, this is exactly where computer simulations became so useful, as they allow to conduct laboratory research promptly and effectively to support and improve clinical practice almost immediately by providing concrete conclusions that can be translated to the clinical setting. Thank you very much. My contact information is here, and I'll also be happy to take questions either online or offline if you reach out to me, for example, via email.
Thank you ever so much, Amit. It's really fascinating to see all of your work, and we're obviously very proud to support you with our software and all of the research work that you've been completing. And thank you also everyone listening. It's been a pleasure hosting this webinar today, and we hope you really have enjoyed it. If you'd like to maybe try the Simpleware software for yourself, then you would be more than welcome to do so. The easiest way is for you to go to our website, the address of which you can see at the bottom, so www.synopsys.com/simpleware, and then from there you can find your way to the trial request page. But so thank you once again, everyone, and thank you very much, Amit. I think now with this we will draw to a close and start the Q&A, where we can answer your questions as best as possible. So thank you once again. So hello everyone again. So we're going to start the Q&A now. So I'm just making sure that Amit and myself are unmuted. Amit, fantastic. Thank you very much for the webinar. The questions have started to roll in. So I've got one ready to fire at you straight away. This is the question is the vicious cycle a mechanism that occurs when pressure ulcers are formed via dynamic conditions? If not, how does this vary?
Dynamic conditions is kind of a broad term. Dynamic conditions can refer to dynamic mechanical loading or dynamic thermal loading, or to the dynamics of the physiology, which is ever evolving. Even in what we perceive as a static environment, say of a patient who's lying in bed and does not move, there are these micro movements. There are movements due to breathing or mechanical ventilation. There are movements due to spontaneous muscle contractions. There are movements due to repositioning of that patient in bed, and similar phenomena relate to pressure ulcers that are caused by medical devices. And obviously, once a patient has moved, or when he moves or she moves, the thermal conditions change as well, and the body responds. So this is a very complex system of interactions that I've tried to simplify, but in reality, it really depends on the specific scenario, the clinical scenario. And one other aspect that relates to that is that the body responses and the tissue responses evolve due to the condition, but also in response to the environment. And so there are feedbacks continuously between what we call boundary conditions or interface conditions and tissue responses, which are also manifested by, for example, changes in tissue stiffness over time. If edema develops and the tissue swells, there's swelling pressure, and so the stiffness changes. That obviously interacts with the boundary conditions that change, and so it's an ongoing dynamic process, even if it looks static.
Sure. That actually, it relates to another question that's come in. And that is more specifically focused around the time. Oh no, sorry, it's a different one. So it was more looking at whether the time threshold for tissue damage is something that's stable, or does it change? So the actual question was, is there a time threshold for when tissue damage occurs, or is this very patient specific?
The latter, of course. If there was this magic number, my work would have been very easy.
Sure.
But unfortunately, there isn't. It's very, very individualized. And it's individualized because of all these factors that I've just mentioned in response to the previous question, where I've described a patient-specific and environment-specific interaction, or set of interactions. And on top of that, there is the age, there is the medical condition, there are the medications, there are the treatments, and all of this together, and COVID being just one example of how a disease, an acute disease in this particular example, can affect all these interactions. But you can also think of a million chronic conditions in the background, from, I don't know, from heart failure that affects your vascular performances, to pneumonia, which would reduce your tissue oxygenation, and through diabetes, which would affect your perfusion capacities, particularly the peripheries of the body, but also your inflammatory response, and so on and so forth. it's kind of a rule of thumb that any neuromuscular condition that obviously introduces a risk for a pressure ulcer, for a pressure injury, also affects these other factors, such as the inflammatory system. And again, just to give you an example, the neurological system and the inflammatory system, they're coupled. And the inflammatory response uses the neurological system for its activity as a platform. And so basically everything influences everything else, which is different across the individual patients. And so it's almost impossible to predict, definitely not by using these coarse, these rough measures such as a threshold. You would need to have individual examinations and measures of the tissue status, skin and deeper tissue status, and then you would need to correlate all of this with the risk for injury, but that will always be patient specific.
Mm-hmm. Okay, yeah. So I've got another question here, which is more focused towards the computational modeling side. So it's a question stating, how long did it take to conduct FEA for the wound modeling?
How long does it take to conduct? So there is the phase of developing a model, which takes typically in my research group from several months to a year. And then there is the issue of running it and actually performing the simulation, because once you have a model in place, you would like to do many simulations to explore different scenarios that are relevant to the model configuration. And that, again, depending on your computer software and hardware, particularly the hardware, and we are using very powerful hardware that we need to update roughly every 2 years. Typically several hours, can be overnight, but I've also developed with my group some models that took 2 or 3 days to solve.
Yes, yeah. So significant times, but worthy results to draw some conclusions for.
Well, it's definitely quicker than running a clinical trial, right? So if you would run a clinical trial, that would take months to years. If you have a model that has been developed and has been verified, you know, running it overnight and getting some answers, it's not even on the same scale.
Certainly. And that's actually in line with another question here. So it was regarding some of your concluding remarks. With the pandemic significantly interrupting traditional clinical approaches, clinical trial approaches, do you feel the in silico approach will be expedited or be used more quickly?
It's already happening.
Replace large amounts of in vivo study.
It's already happening, no doubt about it. And we could see that, in fact, prior to the pandemic. There was this existing trend, for example, of replacing animal model studies with in silico work where possible. And my own work has shifted over the years from doing animal studies to doing more lab work that does not use animal models. And then the pandemic brought another aspect of, basically all clinical trials have stopped, both because there were no resources, manpower, etc., and because there was the risk of infections, and so clinical trials, which are expensive and complicated to perform and deliver, have been an issue in terms of the cost effectiveness of biomechanical research for many, many years. But I think that this was a great example that has taught us, much like other things that this pandemic has taught us, that we can use those computer models to understand phenomena and act upon them very fast, and then collect clinical data as we can. I'm not saying that clinical trials are redundant, just to be clear on that. I am saying that with this level of sophistication and excellent computer tools such as the product that you have, but also given the computer power that we have today, I don't think that, at least prior to the pandemic, we have utilized all these capacities prior to starting clinical trials. I think that we need to do more computer modeling prior to doing clinical trials to focus the efforts and to optimize the cost benefit of the trial. And only start the trial when you have a rough idea of what to expect. And I always compare that to the automotive industry. Imagine car companies, motor vehicle companies crushing cars on concrete walls every day. How soon can we expect that they will go, into bankruptcy? And so they do, and that's the logical thing to do. They do 1 million simulations before they would crash a car. Because crashing a car is, it's a classic example of what engineers would say is a destructive test, as opposed to computer simulations where you can run the trial all over again, and change this parameter and that parameter, and you've consumed some electricity and spent some time, but basically that's it. And I think that the same approach needs to really be implemented in the world of clinical trials, probably in any field, but definitely in my field of wound research. There's so much that can be done with computer modeling, and we have been pioneering for now, I would say 20 years and I've seen the field growing, and I've seen these computer software and hardware tools getting better and better and stronger and more powerful. And I think that we can do things now that as a PhD student, where I already at that time did computer simulations in the world of wounds, I did not imagine that one would possibly be, one could possibly do, and we're doing that now.
Yes, yeah, fantastic. It's I guess things that are only a dream merely a few years ago are now a reality for this sort of simulation.
Yeah.
So I guess leading on from that, we've got another question here. So it's asking, did you conduct only in silico experimentation, or was it were some of your tests performed on test models, patients as well in some of the cases?
My research has always been integrated, experimental computational, and in fact, every computer model needs to be validated in some form against real world data, empirical work. And so we are using a variety of tools for that. We are developing and have developed in the past many different tissue, organ, and body phantoms made of, say, synthetic materials, or sometimes even tissues incorporated in these phantoms, say animal tissues, and we have put sensors in those phantoms. Basically, again, using the reference to the car industry. Think of mannequins, crash test dummies, or whatever you want to call them. These are body or tissue phantoms, and we're doing the same thing. Again, there they are testing the likelihood of an injury under impact conditions. We are doing a similar thing in most cases under sustained loading conditions, but the line of thought is very similar. So this is a very effective way to validate a computer model, though the disadvantage would be that you typically look at point measurements in these phantoms. So you would put sensors in discrete places, in discrete locations in the phantom, and you would take measurements from there, as opposed to the again, the really nice feature of looking at distributions of tissue loads and then understanding how the load transfers from one place to another in the structure and tissues and the body as a whole is a structure. So the huge advantage in doing computer simulations is that you really look at how load transfer is being conducted within the tissues, or sometimes even at the micro scale, because we have done these things on cells as well, individual single cell models or models of groups of cells. I've shown some of that in the talk. So the validation is essential, and eventually, you need to go to some kind of a living model system. We've used a lot of imaging for that, MRI, low dose CT, ultrasound. We've worked with all these imaging modalities to say quantify tissue deformations in vivo in actual patients. And there are other types of living model systems that, for example, give you an idea about the tissue tolerance under certain conditions to mechanical loading. We work with that as, I see the world as a big puzzle, and I can only look at one piece at a time, but at certain points, I try to combine the pieces together to tell a story, which is what I did today. But an essential part, a critical part, a vital part of this story would be the validation aspects and the experimental work that is needed for that. And again, these are the main tools that we are using: body and tissue phantoms and living model systems.
Sure, certainly. And if you don't mind, a question from myself regarding the phantoms. It's an interesting area. I wonder, how do you manufacture those yourselves internally within the group, or do you
Yes, so most of them we manufacture ourselves, and over the years we've developed expertise in that as well. But sometimes we would buy certain material components, such as, for example, materials that are used for training surgeons or physicians in general for simulating tissues. So we don't necessarily invent the wheel. I'll just give you an example. We're working with skin now, and obviously we're using animal skin samples, but that's not always convenient when you want really robust data, kind of taking out the biological variability aspects. And then we would, in places, use skin simulates that have been developed and are commercially sold for training surgeons on how to suture. Yeah.
Okay. Yeah.
There's
Okay,
a world
I see,
of possibilities.
Yes, and we're aware of groups that we work with, certainly, that are using 3D printing techniques as well to manufacture certain
of course,
yeah, biological structures that are mimicking the properties of those tissues that they represent too. So it is a fascinating area, certainly. Okay, Amit, I think that's we've come to the end of the questions that have been rolling in now. So I think what's left is for me to say thank you very much again, and we really appreciate your time. It's been a really great talk that you've given us today, and it's always good to see all of the work that you're completing and how you're implementing the software and computational modeling in these really interesting ways to provide such great feedback to the clinical side of the clinical approaches to addressing wounds and caring for those people. So thank you very much once again.
David, thank you. And I'd like to also thank the entire Synopsys team for the opportunity and for all the logistics and arrangements, and enjoy your time in the garden. And I wish everybody good health and take care.
Likewise, likewise. Thank you very much. And I think I believe Matt's still on the line, so I don't know if there's anything on the technical side we need to do to wrap up the webinar and make sure everyone's aware of what happened next. But I think I can speak to that a little bit. So everyone listening, the webinar will be available for you online to watch again and refer back to, and all of the material will be available to watch there. So I'm sure you'll be sent the link as well following the webinar, so you'll be able to access it very readily. Okay, so thank you very much again. And I think that's it. We've come to the end of the webinar. So thank you again.
Bye for now.
Speak to you soon.
Bye-bye.
Bye-bye.